Assalamu alaikum viewers, I hope you all fine. Today our topic of discussion is DNA barcoding. So let's start. Bismillahirrahmanirrahim. DNA barcoding. These are the contents that we discuss one by one. What is DNA barcoding? DNA barcoding is a fast and accurate system to identify species by using short DNA sequence instead of whole genome. This short DNA sequence is generated from standard region of genome that is known as marker. This marker is different for various species like CO1, Cytochrome C Oxidase 1 for animals, MATK for plants, and Internal Transcribed Spacer for fungus. DNA barcode is usually made of 4 nucleotide bases: Adenine, Thymine, Cytosine, and Guanine. Each base is represented by a unique color in DNA barcode. As in this slide, adenine is shown in green, thymine in red, cytosine in blue, guanine in black color. Cytochrome C Oxidase 1, which is present in mitochondrial gene in animals, that is often regarded as official marker for animal. It is proposed by Paul Hebert and recognized by International Barcode of Life. It is suitable for these group of organisms because it is uniform in them, and it is not suitable for other group of organisms. So, Internal Transcribed Spacer for fungus and 2 genes like RBC I and MATK from chloroplast genome are recognized as barcode markers by IBOL for plants. History. It first came to the attention of the scientific community in 2003 when science research group of Paul Hebert published a paper titled Biological Identifications Through DNA Barcodes. So therefore it is a new tool for identification of species and for taxonomic research. Procedure. The process of DNA barcoding involves 2 basic steps. First step is to build a barcode library of identified species. Second is matching the barcode sequence of the unknown sample with the barcode library for its identification, that is known as sequence alignment. The first step requires 1 or several individuals per species as reference samples in the barcode library. Tissue samples can also be collected from live species in field or from specimen in museum for generating. These specimens go through lab processes that are tissue sampling and DNA processing and sequencing to generate DNA barcode in form of chromatogram. Chromatogram is visual representation of DNA sequence produced by sequencer. In the last step, this barcode can be stored in database for future use or can be used as query sequence to be compared with sequence already present in database. In this slide, chromatograms show the visual representation of DNA sequences that are produced by sequencer. Grammatically, you can collect fresh specimen or preserved specimen from museum, and these two specimens go through lab process, that is tissue sampling and DNA extracting and sequencing to generate DNA barcode in form of chromatogram that is produced by sequencer. In the last step, this barcode can be stored in databases like IBOL, GenBank to use in the future, or can be used as a query sequence to be compared with already stored sequences to check their duplication. Importance: global climate lead to importance of identification of organisms to preserve species because of increasing habitat destruction because of biological effects. About 5 to 50 million plants and animals are living on Earth, out of which less than 2 million have been identified. Extinctions of animals and plants is increasing yearly, means thousands of them are lost each year, and most of them are not identified yet. This destruction and endangerment of ecosystem has led to an improved system for identifying species. In recent years, as I told you in previous slide 2003, new ecological approach called DNA barcoding has been proposed to identify species and ecology research. So that's why DNA barcoding is so important to identify species that are not identified yet and for ecology research. Applications. First one, controlling agricultural pest. DNA barcoding can help in identifying pests in any stage of life, making easier to control them and saving farmers from cost of $billions from pest damage. Second one, identifying disease vectors. DNA barcoding allows non-ecologists to identify the vector species that cause serious infectious diseases to humans and animals and understand these diseases, their mechanisms, their treatments, and cure them. Third one, sustaining natural resources. Natural resource managers can monitor illegal trade of products made of natural resources like hardwood trees by using the DNA barcoding. For example, Fish-BOL is reference barcode library for hardwood trees to improve management and conservation of natural resources. 4.1 Protecting endangered species. Primate population is reduced in Africa by 90% because of bush meat hunting. Overall population is also reduced due to global warming, greenhouse effect. DNA barcoding can be used by law enforcement to bush meat in local markets, which is obtained from bush meat. Next, monitoring water quality. Water is a process resource for living being. You can measure the organisms' health that are living in lakes, rivers, and streams. DNA barcoding is used to create a library of these species that can be difficult to identify. Barcoding can be used by environmental agencies to improve determination of quality of water and to create better policies which can ensure safe supply of drinking water. Next, routine authentication of natural health products. Authenticity of natural health products is an important legal, economic, health and conservation issue. Natural health products are often considered as safe because of their natural origin. So that is routine authentication of natural health products, because natural products are safe to use because of their natural origin. Next, identifying of plant leaves even if flowers or fruit are not available. Next, identification of medical plants. Conclusion: DNA barcoding is a system for fast and accurate species identification which make ecological system more accessible. It has many applications in various fields that are we discussed, like controlling agricultural pest, protecting endangered species, sustaining natural resources, monitoring water quality. And these are the references. Thank you so much for listening. Allah Hafiz.